Hemoglobin. The quaternary structure of a typical hemoglobin molecule is 4 subunits, Hb4, 2 alphas and 2 beta subunits, and each hemoglobin subunit carries one oxygen molecule, HbO2. Then Hb4 will carry 4 oxygen molecules, Hb4O24, as it passes through the systemic capillaries. Usually, hemoglobin only gives up one oxygen molecule, and so Hb4O24 becomes Hb4O23 and one free oxygen molecule which diffuses in solution through the capillary wall into the interstitial fluid of the surrounding tissues. So Hb4O24 represents a normal arterial oxygen saturation of approximately 100%, and Hb4O23 represents a normal venous oxygen saturation of approximately 75%. Logical, isn't it? Hb4O22 represents a profoundly low oxygen saturation of approximately 50%, and if this is arterial blood, then your patient is near to death. Further, a true mixed venous blood saturation of 50% tells you that the patient has an extreme metabolic emergency, indicating very severely inadequate arterial oxygen supply. Hb4O2 cannot occur in physiological conditions. The hemoglobin needs to be physically denatured to drive off the third oxygen molecule to desaturate it down to 25%. So the third and fourth oxygens are essentially never available to us for metabolism. Well, let's return to our journey. The blood in the radial artery will pass down the arterioles and into the capillaries, which serve, for example, the muscles of the hand and fingers. The cells of the hand and fingers are alive, they're living tissue, so even at rest they will have significant demand for oxygen. But if the muscles of the hand and the hand and fingers are working hard, such as when you're in the airport carrying heavy luggage around and you have to keep going because you've got to make a connection, then their metabolic rate will be very high because the muscles are working, and so will their demand for oxygen be very high. So this slide represents the millions of cells as little white circles, and each contains a little green mitochondrion. So these represent the muscle cells in the fingers. Many of you will know that I love everyday analogies, and this situation is very like a busy factory at the peak of production. There is unfortunately the unavoidable production of waste products, represented by the smoke plumes issuing from the tall chimney stacks from this factory. Now, recall, we discussed earlier how hemoglobin absolutely will not release its oxygen unless it receives very specific and precise signals to do so. We call these precise signals triggers for the release of oxygen. And these triggers are metabolic byproducts in living tissues, which are in the living tissues that are very metabolically active, just like the plumes of smoke from a factory. Do you remember what these triggers are? The metabolic triggers for oxygen release are the end products of intense metabolic activity, which is in fact what is generating the need for oxygen. The triggers are increased concentration of hydrogen ions, or acidemia, or increased protons, an increased partial pressure of carbon dioxide, an increased concentration of 2,3-diphosphoglycerate, and an increase in local temperature. 2,3-diphosphoglycerate is a side product of glycolysis and is an excellent marker for increased metabolic activity in glycolysis. And as soon as hemoglobin sees these triggers, the protons and the CO2 combine with a hemoglobin subunit to change the stereochemistry between neighboring hemoglobin subunits. It's a subtle change, but functionally it's a very important biochemical change. Here we see an image of a hemoglobin molecule in a red cell in an arteriole. Here we have hemoglobin arriving in the capillary, and it has a high affinity for oxygen. The mitochondria in the cells of the tissue are highly metabolically active and are consuming a lot of oxygen. the well-oxygenated hemoglobin is now moving into an environment with a low partial pressure of oxygen. The red cells are about to pass close to a region consuming a lot of oxygen and producing a lot of triggers which decrease hemoglobin's affinity for oxygen. These triggers decrease hemoglobin-oxygen affinity, and so a single oxygen is released from one hemoglobin subunit. If oxygen continues to diffuse off the hemoglobin and move into the tissues, the partial pressure of oxygen in the capillary has to decrease because it's losing oxygen. Hemoglobin now has only a low affinity for oxygen, which is free to diffuse off the hemoglobin in the presence of an appropriate oxygen partial pressure gradient. This change in one subunit decreases the affinity of oxygen in a neighbouring subunit, and so an oxygen molecule is released into the blood, where it is now in solution and it is free to diffuse down any oxygen partial pressure gradient that exists, which of course takes it towards actively metabolic cells, which are those in fact that have the greatest need for the oxygen. So, what's the partial pressure of oxygen at this location? You probably don't have a number that you can immediately recall, but of course, we can work it out. Do you know the partial pressure of oxygen in venous blood? You may have learned this in your respiratory physiology, or you may have seen it on a venous blood gas form. So, imagine the oxygenated hemoglobin entering a tissue capillary, supplying exercising muscles in the fingers and in the hand. And as this molecule encounters the specific metabolic triggers, it starts to release its oxygen, and the oxygen diffuses off more and more hemoglobin molecules are giving up and losing their oxygen molecule. But when the oxygenated hemoglobin encounters an environment where the partial pressures of oxygen in the tissue is exactly the same as the partial pressure as the oxygen in the capillary, the oxygen is now free to diffuse off the hemoglobin, but it will not do so because there's no partial pressure gradient between the capillary and the tissue fluids. The partially deoxygenated hemoglobin, HbO2/3, will then remain in the capillary and enter the vein. So the partial pressure of oxygen on hemoglobin entering the venous blood, which is normally 40 millimeters of mercury, must be the same partial pressure at which no more oxygen will leave its hemoglobin. So this must be the partial pressure of oxygen in the tissues. This is referred to as the equilibrium partial pressure between oxygenated hemoglobin in the capillary and the oxygen tension in the tissues. And for tension, we mean partial pressure. The terms tend to be interchangeable. As a quick digression, note that an oxygen partial pressure of 100 millimeters of mercury is more than adequate to generate 100% oxygen saturation in arterial blood. But a partial pressure of oxygen of only 40 millimeters of mercury in venous blood is adequate to generate a venous oxygen saturation as high as 75%. Yes, 75. And this, of course, should remind us that the relationship between oxygen partial pressure and the resulting oxygen saturation of hemoglobin is not linear. In fact, we all know this. We know that the oxygen dissociation curve for hemoglobin is a sigmoid shape. The ability of hydrogen ions, carbon dioxide, 2,3-diphosphoglycerate, and increased temperature to influence the affinity of hemoglobin for oxygen is really very important. And it acts through an allosteric effect at each hemoglobin monomer's binding site for oxygen. And you probably remember reading all about this in your biochemistry textbooks under the topics of allosterism and cooperativity. The effect of CO2 and hydrogen ions on binding oxygen is called the Bohr effect, named for a famous Respiratory Physiology, Christian Bohr, shown right here. And he's actually the father of the famous Niels Bohr, who discovered and described the quantum physics. The opposite effect occurs in the lung, where binding of oxygen drives off hemoglobin-bound carbon dioxide. And this opposite effect is called the Haldane effect, named for Sir John Scott Haldane. And both effects are mediated at a biochemical level by a phenomena called cooperativity, due to subtle changes of oxygen on CO2 binding, and conversely, CO2 on oxygen binding by an effect on adjacent hemoglobin subunits. It's fascinating, incredible how hemoglobin works. So again, let's return to oxygen's journey. Oxygen in the interstitial fluid around the capillary is at approximately 40 millimeters of mercury partial pressure, but this of course varies highly between individuals and with different levels of tissue metabolic activity. But we can be sure that by the inverse square law, as the distance from the capillary decreases a little, the prevailing oxygen, when the distance increases a little, the prevailing oxygen tension decreases a lot by the inverse square law. The oxygen will diffuse through the cell membrane, and the partial pressure of oxygen inside the cell, once again, is highly variable. But it's probably of the order of about 20 millimeters of mercury-ish. And then the intracellular oxygen will diffuse from the cytoplasm into the mitochondria, where the partial pressure of oxygen needs to be about 5 to 10 millimeters of mercury. At less than 2.5 millimeters of mercury oxygen partial pressure, cellular respiration in the mitochondria will cease. So let's think about this. This is very important and very relevant. Life for us aerobic heterotrophs, that is, like mammals, aerobic heterotrophs, life is acidogenic. We produce toxic waste as hydrogens, that is, protons and electrons. In the mitochondria, the Krebs citric acid cycle chops carbons from C6 and C5 intermediates in the cycle, releasing them as carbon dioxide, with the energy of the bond captured by nicotine-adenine dinucleotide H+. The NADH+ then releases one proton and one electron to the electron carrier chain. As the electron passes down each step of decreasing potential energy in the carrier chain, energy is released and is stored by pumping protons. The buildup of these positively charged protons in the intermembrane space eventually exceeds a threshold, and they must return in a rush through the only channel available to them, which is the inner channel of the ATP synthase complex, which is forced to rotate, actually like a turbine engine. And this energy is trapped by converting ADP and inorganic phosphate into high energy ATP. At the end of the chain, the electrons have got to be removed or the whole thing shuts down. If they're not removed, the entire process of oxidative phosphorylation fails, and the accumulation of protons must also be dealt with. They cannot be allowed to accumulate to too high a level. So, an oxygen molecule at the end of the chain is available to combine with 4 protons and 4 electrons to become 2 water molecules. So, please indulge me for a moment. It's imperative to acknowledge the genius of this system. Being able to use oxygen, what was originally a potentially toxic gas, to release the energy of chemical bonds in food, while simultaneously eliminating the waste products of protons and electrons by converting them all into a much needed precious substance, life-sustaining water. This is just a phenomenal achievement. Think about it, it's very important. How moving is this, that life devised a way to be so very clever to promote the continued existence of life on the planet Earth. Quite phenomenal. So I hope you agree. This really is a profoundly important point. The oxygen we breathe in is used to help keep the citric acid cycle and oxidative phosphorylation running. And this oxygen is converted into metabolic water, which over the course of a day accounts for the production of about 500 mL of metabolic water. In an average human being, 500 mLs is not a lot. It's diluted in a total body water of 42 litres. So if you have a person who breathes in radioactive oxygen, many of my students assume that we breathe out radioactive CO2, but this is just not the case. The radioactive oxygen reappears in water molecules created in the final step of oxidative phosphorylation. Of course, the 500 mL of water is diluted into a pre-existing volume of 42 litres. So, of course, it's going to take a long time to appear in the urine, but it will eventually. Oxygen is converted to water, and water is precious to the organism. But the oxygen atom in metabolic water has come ultimately from the atmosphere. And so let's recap this important journey, this saga of oxygen. So the oxygen cascade is, in summary, a partial pressure of oxygen of 150 millimeters of mercury in the atmosphere, approximately 105 millimeters of mercury in the alveoli, 105 in the pulmonary and capillary blood, 100 in blood leaving the left ventricle, 100 millimeters of mercury in blood arriving at the tissues in the arterioles, a partial pressure of 40 in the interstitial fluid, and the same 40 millimeters of mercury in the venous blood leaving the tissues, a partial pressure of very approximately 20 inside the cells, and approximately 5 to 10 inside the mitochondria. Again, these are representative numbers, but they can be very variable from individual to individual and from different metabolic states. The partial pressure of oxygen of 100 millimeters of mercury generates an oxygen saturation of 100% in arterial blood, but a partial pressure of oxygen in venous blood of only 40 will generate an oxygen saturation of 75%. And this is not intuitive because of the unique, non-linear shape of the hemoglobin oxygen dissociation curve. It's a sigmoid shape, not linear. Now think about what we have discussed in this presentation. You've probably heard all this before. The role of the bronchial veins and the Thebesian veins, and the triggers for oxygen release from hemoglobin. Many of you knew all this stuff, and yet many of you will have had difficulty in recalling it in order to answer the questions I asked you. You hadn't forgotten it, but this information was unavailable to you when you needed it. Why? The explanation is this. You probably learned these physiology facts in a vacuum, reading them from a textbook, with no clinical context or relevance. And so you learned them without a true understanding of their real significance for patient care, for taking care of critically ill and unstable patients. Pure physiology can be taught by academic researchers, but I strongly feel that applied physiology must be taught by the clinicians who use it on a daily basis. Now, just a note. The term airway, used by an anesthesiologist like me, is a broad term. It is the oxygen pathway from the anesthesia machine to the alveolus, or from the atmosphere to the alveolus. And it has been said with some appropriate gravity that the anesthesiologist must worship at the altar of the airway, because the airway is such a major priority. So why is all this physiology stuff so critical for the anesthesiologist to know? It's because the administration of anesthesia can jeopardize this very process of oxygen transport at every level. At the level of the airway, in the lungs, in the cardiovascular system, and in the microcirculation. And if we have the arrogance to intentionally compromise a system that is working, we must have the abilities that are required to correct the problems we've caused. And we do this on a daily basis in the operating room. Remember, the practice of anesthesia is a very hard taskmaster, and mistakes are punished very, very, very severely. It is a very unforgiving specialty. We must never lose sight of the fact that other people's lives, our patients, are incredibly important, and our mandate to do no harm is, I believe, a sacred duty. This is why we train for 12 years at least, 4 years at college, 4 years at medical school, and 4 years of residency, just to be worthy of the trust our patients put in us. Thank you for listening to my presentation. You have reviewed many familiar facts and learned some new ones, but I hope you have learned something much more important, a deeper understanding of physiology, hopefully taking your knowledge of physiology from the Bachelor of Science level to the PhD level, giving you a whole new way of thinking about the cardiovascular and the respiratory systems, and giving you new insights into oxygen and the amazing saga of its journey, a journey from atmosphere to ATP synthesis. Remember, in the practice of medicine in general, and in anesthesiology in particular, knowledge is power. This knowledge empowers you to be an infinitely better and safer and more effective physician. Thank you for listening to my presentation. I hope it's helped you.